Welcome back to Champion Medic. This is AQA Biology Unit 1, June 2010. We recommend doing the paper before you watch this walkthrough.
So, question 1A, simple knowledge recall from GCSE, where we asked what are the three structures found in the prokaryotic set of cells, but not in eukaryotes. So, one of the most obvious ones is a slime capsule, which is the outer, like the first outer layer of a prokaryotic cell. Also, non-linear or circular DNA. That's a distinct feature because we know that in eukaryotic cells that DNA is found in the nucleus, whereas in prokaryotic cells it's found in plasmid or non-linear slash circular DNA. Another point is the difference in ribosomes between eukaryotic cells and prokaryotic cells. So in prokaryotic cells you have 70S ribosomes, whereas in eukaryotic cells you have 80S ribosomes.
That's just the difference in size, so ATS is larger.
Yep. And much more dense in terms of size. We can also label rest of the diagrams as well. So
So we
we've
have a
got
flagellum.
mitochondria. The flagellum
Yeah.
on. Yeah,
There's also these, I think they're pilus.
pilus. Yeah.
They just kind of stick out at the end of the bacteria. Okay, part B.
So it's asking about water potential, and that strongly links to the ideas of osmosis. And so the angle that we need to take is if you have the entry of chloride ions into the small intestine, then you're going to have a lower water potential in the lumen. And as a result of that, you're going to have water entering the lumen or the intestines by osmosis. Osmosis is a key word there. And the funny thing is, this question came up in the last paper, didn't it?
Yeah, it did. So that just
And
shows you need to.
yeah,
It's basic, but you don't want to get it wrong in the exam.
so it basically tells you this type of question is going to come up every single year. So it's a concept that you should learn. Yeah, okay, so next question. People with diarrhea suffer fluid loss. Okay.
So this question's
Usage
about
of,
oral rehydration solutions. That
yeah.
is no longer on the spec, but they could ask you to kind of work it out in the exam. So you don't need to know it specifically, but you need to know the concepts behind it.
Yeah, so essentially it can come up as a application question. So in the application skills knowledge section, so I'd recommend you kind of have a general idea of what a oral rehydration solution is, but you don't need to know it in complete detail. Why do you need to boil the water? That's because boiling water helps kill the bacteria or kill microorganisms. Make sure that you, in any kind of question that you're always being specific, whether it's bacteria or the word pathogens. Do not use words like kills germs because that's quite unscientific for an A-level kind of paper. The next question asks, give another type of substance that has to be within the ORS. So they're looking for a type of set of ions. So you can say sodium ions, or you can say potassium ions, or chloride ions.
The reason is because they need to lower the potential, the water potential in the lumen, so that water will go back into the cells.
Yeah. because people with the diarrhea suffer from fluid loss, as it says on the first line paper. So you need to replenish that water supply. All right, question number 2. So you can see a graph similar to what we've drawn on your piece of paper, and it shows the volume of air in a person's lungs when they're breathing. And the first question, they want you to give a simple explanation why there's that change in breathing between points A and B, and that's simply because the lung volume increases. It's literally stated within. If you look at the graph, you can see it really clearly.
You can see it right here.
Yeah. Next, they want you to describe and give an explanation why the shape of the diaphragm is as it is.
So we've drawn diagrams so that you can see what happens when the diaphragm relaxes and is contracted. So the first diagram, the diaphragm, which is this here, it's relaxed. That's why it's got the dome shape. So diaphragm is relaxed, and that decreases the thoracic volume, which means there is a lower pressure inside the lungs, causing air to rush outwards. Sorry, there's a higher pressure inside the lungs. So this
Yeah.
has high pressure, and air will rush out.
And because of the higher pressure, it's higher pressure because you have a lower volume of air in the lungs.
So low volume. You can refer to it as thoracic volume, so it's the volume in the thorax. And then the second one,
Which is the one that applies to this situation, this question specific.
so the diaphragm has contracted into a flattened disc shape. It's kind of counterintuitive, but just remember that when you breathe out, your chest moves down and in. So that will be the first one. The first one's breathing out. The second one is breathing in. Your chest moves up and out, so your diaphragm flattens. That will decrease the pressure because you have a higher thoracic volume. That will cause air to rush in. Okay.
Yeah. So for this question in specific, you know that the diaphragm is going to contract and then therefore flatten or move down because of the because of the increase in thoracic volume.
All you have to say for this question.
Yeah, and if ever in an exam you don't know, just like try breathing in it, like practice.
Practice breathing.
Practice breathing.
So describe how the graph shows the pulmonary ventilation change. The first thing to note is the equation. So pulmonary ventilation is the tidal volume times the breathing rate. So then if we go up to the graph, so I'll quickly write that down in short form. Tidal volume times breathing rate. It's asking between C and D, these two. The breathing rate will increase. We can see that because there are more breaths per minute. If we just look at how much more closely together the lines are packed, the peaks get closer together. and the tidal volume increases, we can see the volume of air in the lungs gradually increases with each peak. And we can use those two sets of data, so the tidal volume and the breathing rate, to calculate the pulmonary ventilation, which is therefore increasing if both of these two factors are increasing. So this equation is already 1 mark. Then you say the breathing rate increases. And the tidal volume increases. Or the volume of air per breath increases, you could say. And then these two, you should also give some data from the graph, or you could say you could give a calculation or something just to prove that it increases. So we'll leave that up to you. Here's we'll just write calculation, and that'll be the 3 marks for that question.
Yeah, it's relatively simple knowledge to recall. But the peptide bond is the middle bit, isn't it?
Yeah, this is the peptide bond here. This on the left is the amide group.
Amino group.
See?
Yeah, it's the amino group.
And on the right is the
carboxyl group. Carboxyl group, yeah. And then you got your R group at the top. So that's what differentiates all the different types of amino acids from each other. I have seen it come up in past papers where they do ask you to draw the structure of how a standard amino acid would look like, so I'd recommend learning how it roughly looks like as well. So part B, it's asking us to show the passage or the order in which an amino acid is guided out of a cell. So we start at F, which is the cytoplasm, and then from there on we go to H, which is the rough endoplasmic reticulum, or RER. It's called the rough endoplasmic reticulum because it has ribosomes on the surface of it, and those ribosomes are involved in protein synthesis. After H, the protein is then transported into J, which is the Golgi apparatus, and at the Golgi apparatus, it processes and packages new lipids and proteins. And then from J goes to E. E is a part of J, but it's known as the Golgi vesicle. So the Golgi vesicles are fluid-filled sacs, which are surrounded by a membrane. And what happens in the vesicles is that it stores the lipids and proteins which are made in the Golgi apparatus, and then it transports them out of the cell to organelle K, and K is the cell membrane. From where on is transported out of the cell into the body.
By exocytosis.
Exocytosis, yeah.
You'll notice G isn't involved because that's the mitochondrion. So that isn't involved at any point of this process. It's purely, mitochondrion is purely there for aerobic respiration, for which an amino acid isn't needed. Part C.
Why are there a lot of organelle G in this cell?
So G we've identified as the mitochondrion.
Mitochondria. And we know that the mitochondria are the site of aerobic respiration. Aerobic meaning with plenty of oxygen. And as a result of that aerobic respiration, you get the release of ATP energy. And that ATP energy is important for
active transport.
The process of active transport, which takes place against the concentration gradient, which is why you need the energy. the ATP-NG. Make sure that you use the word release, because I kept on making this mistake of writing the word produces or makes energy when it actually only releases the energy, because the mark scheme tells you to reject words such as produce or makes, because I guess it's not it's a bit vague, so just be a bit specific by using the word release ATP-NG. So the next part is talking about three
Self-fractionation.
sets of factors. Yeah, self-fractionation and three sets of factors. And they're asking for the reasons why the scientists carried out each step. So, part one, why did the scientists homogenize the tissue? That's to break open the cells. So, and then why, the next part, why did they filter the resulting suspension? Filtering takes place to ensure that you remove or take away or filter out any cell debris, which are excess unwanted parts of the cell.
Or anything else that's in
After
there, like a tissue or any
definitely, yeah.
complete cells.
After that, why did they keep the suspension ice cold? And that's to prevent enzyme activity or enzyme reactions taking place.
Those enzymes, if they didn't put it in ice cold conditions, the enzymes would digest all of the organelles that they're trying to extract. So this prevents that.
Yeah.
And then why are they in isotonic solution?
That is to prevent the process of osmosis taking place, because you want to ensure that you don't want water moving in and out of the cell, or in and out of the organelle, sorry, because otherwise it would just cause damage, and you don't want the organelle to burst or shrink in size.
Yeah, so we recommend learning all of these 4 points because we've seen this question come up time and time again, and you don't want to be caught out in the exam. So just memorize these 4 points.
Yeah, I've seen it come up as a 4 marker, but in this instance, they've just done as a 1 mark question. So yeah, I'd recommend that you guys learn it
On
off well.
on the right over here, we've got this is how it would look when they do it. So the red arrow is showing that as you go down, the heavier they get, the heavier the organelles get. Um, so obviously, once you get to the end, you'll have so the nucleus will become a pellet. It will be very like the toughest thing at the bottom. And as you go up, you get the mitochondria, the lysosomes, the endoplasmic reticulum, and the ribosomes.
Yeah. Um, that's for a animal cell, but in plant cells, because you have chloroplasts as well, the chloroplasts come out after the nucleus, but before the mitochondria, so it's in between there. But I'd recommend that you guys learn the order in which it comes out, in which the different organelles come out, just because they could ask. Right. So question number 4. So question number 4 is a evaluation kind of set of questions, and you have to interpret a bar chart, as you can see on your paper, and it tells you that a local newspaper has published the article that HPV, which is the name of the virus, causes cervical cancer, and does the data in the graph support this claim? So here you want to try and provide a balanced argument because it's a 3 mark question. So the first thing you can say is yes, because many people, or many women, sorry, with that have cervical cancer have the HPV 16 virus, because you can see that around 65%, I think, or 66% of women have the HPV-16 virus, make sure you're really specific with the word HPV-16, because your next point after is going to be against the local newspaper, where you're going to say no, because very little women with cervical cancer have HPV-6 and HPV-11. So if you look at the bar chart again, you can see that less than 2% of women have either of those types of viruses. So that's a point against them. You also need another point against, or an explanation why, because of that correlation does not mean causation idea that we mentioned in our last paper, where it's that it could be caused by another factor. Yeah.
Okay. You could give an example also, but it's not necessary. You could just say there could be another factor.
And you could also mention that the scientists haven't set up a control group. So what are they essentially comparing it to? They've got no level of comparison to make, because there's no control group of people that they're testing against.
There's many other things you could say as well, like they didn't study the HPV in healthy women. They didn't study all types of HPV, and it doesn't add up to 100% also. So if you just look at the graph, it doesn't add up to 100%. So we're not really sure about other types of data that exist. Okay, part B.
What is a HPV antigen? So we know an antigen from our general knowledge that is a type protein, and that we know that that protein stimulates an immune response. So even though it says what is a HPV antigen, you don't need to be specific by referring to HPV. Just give your general definition of what an antigen is for 2 marks. So a protein gets you 1 mark, and stimulates an immune response is your other mark. So the next question is asking about the importance of memory cells, and why they're important. So we know from our own knowledge that memory cells are produced, and they remain within the body from the first initial infection, which is the primary response. So we know that the secondary response is going to be much more rapid, and we're going to see a higher rate of production of antibodies in the secondary response, because of the memory cells. And as a result of that, that you can produce more antibodies, you're able to destroy the antigen or the virus before it can create more symptoms, and that's important. So it's a general set of knowledge recall, but it just needs to be applied to this situation.
Again, memorize this answer. You don't want to get caught out in the exam. So this would be the first mark. Those are where all the marks would get are. And it's out of 3, but just give the 4 points just in case they don't accept one of them for any reason.
Part C, what's the advantage of vaccinating men as well as women with the HPV vaccine? So the main advantage is that it prevents males being carriers of HPV, or it destroys HPV as a disease completely in males. Avoid using the word prevents males catching HPV, because it's specifically saying why it's important to give the vaccine specifically to males only. And also the second advantage is it prevents males passing on the virus to other females. Because you want to prevent the spread of this disease and keep it as limited as possible within the population.
Okay.
So question 5 is a general. Although we've been given a lot of information, it's just a general enzyme question. So first, I want to say that as a specific active site, the active site is specific, and as a result of that, it's complementary in shape to the substrate.
If this is the ganglioside, the active site of the hexosaminidase, if that's how you say it, the active site is complementary
Yeah.
to it, which means it can only be broken down by that. So it will form an enzyme substrate complex. This altogether is the ES complex, and that allows it to be broken down.
I think with this question, they've added in a lot of words that we haven't heard before, such as gangliosides or hexosaminidase, but the theory behind it is the same. There's no change, so it's a common answer there to just that you guys just learn off by heart.
It also write forms the ES complex.
Just make sure you guys write out enzyme substrate complex, but we're just doing shorthand for
Yeah.
now.
Whenever you see enzymes, just think of this diagram and it will all make sense.
Right, the next question is referring to Tay-Sachs disease, and it's asking us to show the results that we would first expect with a person with the Tay-Sachs disease. So we should expect a horizontal line.
So if I just draw that, and it will start from where it says the initial is. So that'll be the first one.
And the reason why it's a horizontal line and not another line is in the key part of the question, where if you look at your paper, it says that people with Tay-Sachs disease do not have this enzyme in their blood, which means that if they don't have this enzyme in their blood, that means that enzyme substrate complex reactions cannot take place or cannot be enzyme substrate complexes cannot be formed, which means that the ganglioside concentration is going to remain constant throughout the test. There will be no change from the initial point of adding all the way to the end. So the next question, part 2 of B, is linking to that, asking us what we would expect for a healthy person who doesn't have the disease. And we're going to label this curve H, and we're going to see a decreasing curve, and then more slowly kind of decrease. And the reason why is because initially you're going to have a lot of active sites available for the substrates to bind to, but after that all the active sites are going to get occupied, and which results in a slower rate of reaction, which is why you see the curve not decreasing as rapidly afterwards.
Okay.
So part C is an application question, but it asks why can't we give the enzyme as a tablet? And the reason is because we know that enzymes are proteins, so that's going to get you a first mark. And then after that, we know that proteins can be digested or destroyed by enzymes. So That's the issue. They're going to be destroyed too early on, which means that you're not going to get a chance for the enzymes to react with the substrates to form an enzyme substrate complex.
I
You
prefer talking about it being destroyed by the acid in the stomach because it's a bit more. It's easier to think about because if you take the tablet, it will go to your stomach and then the stomach acid will just break it down.
Right, so question number 6. Make sure you guys read the passage before you go through this. So part A wants us to identify 2 risk factors in relation to coronary heart disease. So one of the most common ones to write is smoking cigarettes. That plays a key part, as well as drinking alcohol. You could also mention age, obesity, a lack of exercise. These are all common answers um, for a risk factor with coronary heart disease.
Yeah, there's all sorts. You could also put gender or genes or stress. There's lots you could put down.
Genes can be traded out for family history. Yeah, so there's a lot.
Part B.
Why are we using healthy volunteers in this experiment that the scientists are investigating? The reason is because you need to have people that have their blood vessels that are working completely normally or at normal size. You don't want the volunteers within your experiment to have damaged blood vessels or blood vessels that have a much smaller diameter due to a disease that they may have, because it may affect your experiment. So if everyone had normally, inverted commas, functioning vessels, you wouldn't get a difference, and it would provide you with much more accurate results. Part 2, why are the scientists dividing people randomly into 2 groups? And the reason is because you want to avoid bias. That's a common answer whenever you see why are volunteers being divided into 2 groups randomly. Why are these plants being divided randomly? The answer is usually avoids bias.
C. Part 1.
Describe how the control group is being treated. You want the control group to be treated exactly same as the experimental group, but there's one difference, which is that you're going to give them chocolate without the flavonoids, I think, if that's how you pronounce them, as it says in the passage. You want them to give chocolate without flavonoids, and the reason is you want to just prove that nothing like the placebo effect is going to happen or something like that.
Whenever you get a question about how a control group should be treated, always start with they should be the exact same as the experimental group, and then give the one difference that there would be. So in this case, the chocolate with without the flavonoids. Sorry, that should say. Whereas the experimental group will have the flavonoids.
Yeah, like the control group questions always appear as 2-mark questions, so those are the 2 points. So just apply it to the situation.
Part 2.
Why is it important? Like, what is the importance of having a control group? Is to make sure that the results that you obtain aren't due to another factor or another substance in the chocolate. And unfortunately, with this question, the mark scheme says you have to be really specific in terms of mentioning that it cannot be due to another substance or factor in the chocolate. You have to mention the word in the chocolate, or you have to say to ensure that the results are due to the flavonoids only. Unfortunately, that's just due to the way that the mark scheme is written. So if you just say to show results are not due to other factors, that doesn't get you the mark. You have to relate it back to the passage.
Yeah.
The next question, part D, is asking us why an increase in the diameter of the lumen causes a reduced risk of coronary heart disease. So the coronary artery would always have a wide lumen, and the reason why is because you want to lower the chance of it having a blood clot, and as a result of that, you're going to have a lower chance of an atheroma appearing. So if you look at the diagram, you can see, let's say that was the coronary artery, you can see that the lumen is quite wide. And as a result of that, there's going to be a lower chance of blood clot forming, which means that you're going to ensure constant supply of oxygen and glucose to the heart muscle, to the cardiac muscle, which ensures that the heart can respire effectively.
It also ensures a lower chance of high blood pressure because a wider lumen means there's a greater volume, which also means you have a lower pressure. Okay, question 7.
So 7A. So the way that I'd approach this question is I'd divide it into 4 different processes that we know. So simple diffusion, osmosis, active transport, and facilitated diffusion. That's going to be your 4 key overall processes. So start off simple with simple diffusion. So we know that simple diffusion takes place from, or is the movement of particles from an area of higher concentration to an area of lower concentration. And we know that process takes place down the concentration gradient. So for example, in this question you can give an example which can get you a mark. So an example is small molecules which are lipid soluble can diffuse through the phospholipid bilayer, which is the diagram you can see in the video, and that goes down the concentration gradient. So then now you're going to talk about your second process, which is osmosis. So in osmosis, it's the net movement of water molecules from a region of higher water potential to a region of lower water potential, and this takes place across a partially permeable membrane. And we know that this also takes place down the water potential gradient. So your next process, is going to be active transport. So active transport we know takes place the opposite way. So it goes against the concentration gradient from an area of lower concentration to an area of higher concentration.
I'll just put that over here so it's clearer.
Yep.
That's the exact opposite of simple diffusion. And it goes against the concentration gradient.
Active transport also requires ATP energy, so that point gets you a mark as well. And then your final process is, your fourth process is facilitated diffusion. So facilitated diffusion involves channel proteins and carrier proteins, does it?
Yeah,
It does.
so the yellow is the channel protein and the blue is the carrier protein. Facilitated diffusion is a lot like simple diffusion. The only difference is that because if it's an ionic substance, it won't be able to pass through the phospholipid bilayer due to the hydrophobic heads, I think it is. Yeah, those will repel the
Yep.
ions. We require. So let's say this is an ion, it won't be able to pass through the phospholipid bilayer, but it will be able to pass through the channel protein by facilitated diffusion from a high to low concentration. So there's a high concentration up here.
Yeah, just to correct, it's the hydrophilic head and it's a hydrophobic tail. Just a correction, yeah.
Is my diagram the wrong way around?
No, it's the right way around.
Hydrophilic means it attracts though.
Yeah, the head is hydrophilic.
Oh yeah, okay.
Because that's why the heads are. So on the inside, you can see that the hydrophilic heads are facing inwards because the water and the cytoplasm is attracted to it.
Okay. And then the carrier proteins on the right, those are for active transport. So if we have a low concentration of a substance here, a higher concentration on this side, and we need to transport it downwards into the cell, we need ATP energy. So this substance will attach itself onto here, and an ATP molecule will just be somewhere around here. It will release its third phosphate group, so triphosphate, it has 3 phosphates. We'll release the third one, and some of that energy will be used to move this carrier protein into this position on the right, and then it will cause this molecule to be released into the area with the higher concentration. So that's against the concentration gradient.
So that concept mainly comes up in the co-transport of sodium ions with glucose, that whole topic. Yep.
So they say if you mention the sodium ion and the glucose
co-transport,
that's also a mark.
Yeah, because you're giving an example similar to how with simple diffusion, if you mention the small molecules passing by a phospholipid bilayer as a process, that gets you a mark. So just to summarize, the way to kind of approach this question is by dividing into 4 processes: simple diffusion, active transport, osmosis, and facilitated diffusion. And so the stuff that gets you the mark, so the word simple diffusion gets you a mark. High to low concentration or down concentration gradient, that's a mark. The word osmosis gets you a mark. Low water potential, high to low water potential, that gets you a mark. Facilitated diffusion as a process gets you a mark. Active transport gets you a mark. Low to high concentration gets you a mark. Uses ATP gets you a mark. And the example of sodium ions with glucose co-transport gets you a mark. So this question is relatively easy to get the 5 marks. So if you do get 4 or 5 marks, that's quite good, because it's quite simple knowledge to recall.
Okay,
so 7b, they want us to describe and explain the process of the adaptations of the lungs for diffusion or gas exchange of oxygen in the alveolus and the capillaries. So this question, you have to ensure that you not only describe, but you also give a valid explanation for the reasons why. So we know that as the structure of a lung, so we'll just go briefly through the structure of a lung. So at the top, you've got the trachea, which is also known as the windpipe. And then you've got the central part, which divides into 2 as the bronchus. And then you've got the bronchioles, which are the small branches coming off it. And then at the end of the bronchioles, the red things that we've drawn are the alveolus or alveoli. Don't know which one's plural or singular.
Alveoli is plural.
Yeah, fair enough. One of them's called alveolus. So if we zoom in, on an alveolus, we can see that there's many alveoli or many alveolar walls which provide a large surface area. So that's a key point. Because you have so many of them, you're going to have a much larger surface area. But it's really important. So here, as we are writing, we've just made a mistake here, which is you have to say many alveoli walls. The word walls gets you the mark. So, for example, if you don't use the word walls, you're not going to get the mark. So you have to mention that the walls provide the large surface area. And then we know that surrounding the walls, we have capillaries. So those capillaries are also going to provide you a large surface area because they're thin and they're 1 cell thick, and they stretch out quite a lot. So that's going to give you a larger surface area. And because of these two factors together, you're going to have a faster rate of diffusion. It's important that you mention that it's a faster rate of diffusion. Fast rate of diffusion. Yeah.
Okay.
Right. The next point that we need to know is that the alveolar epithelium, which is the walls of the alveoli, are really thin. So as a result of this, we're going to get a shorter
Diffusion pathway.
Shorter diffusion pathway. And that means you're going to get also a faster rate of diffusion, as we've just said earlier. So the next set of points that we're going to mention is the concept of ventilation. So we did mention this in our last paper, because this concept is like a recurring theme that comes up again and again in AS topics. So ventilation is the replacement of air. So you're going to bring in air with a high oxygen concentration to replace and remove out air with a lower oxygen concentration. And so this helps maintain a concentration gradient. Make sure you guys mention the word maintains a concentration gradient. So as you can see on the diagram that we've drawn. And as a result of this, you're also going to get faster rate of diffusion. So we've just finished that. So to summarize, many alveoli walls get you a large surface area. That gets you a mark. Many capillaries with large surface area, that gets you a mark. Fast rate of diffusion, that gets you a mark. Alveolar epithelium is thin, that gets you a mark. Shorter diffusion pathway, that gets you a mark. Just on that point, make sure that you guys use scientific terms such as alveolar epithelium, because saying cell walls or thin walls, that won't get you the mark because it's not scientific enough for an A-level biology paper. So make sure you guys use words such as alveolar epithelium is thin. Then ventilation gets you a mark, and maintains a concentration gradient also gets you a mark. So as we said in our last video, we strongly recommend that you learn both of the 5 markers that we just went through, because they could come up as a 3 marker or even a 2 marker, and you don't want to lose marks on such a simple set of knowledge recall questions. Yeah, so that's the end of the paper.
Thank you for watching. We'll be posting more past paper videos soon.
So if you like the video, make sure you guys like the video and subscribe to our channel. See you next video.